Hi everyone, welcome or welcome back to Science Breakdown. Today I will do something a little bit different because I am not going to talk about a paper, I'm going to talk about a Nobel Prize. And more specifically, the first AI related Nobel Prize. So no, it's not to thank Google AI for telling us that we can eat one small rock a day, or Midjourney for creating the coolest images of cats, but to celebrate real scientists who have created a program that has concrete application in the biology field. So I don't know if it's a common feeling, but at least I feel pretty detached from Nobel Prizes. I rarely know what is celebrated, and even when I do, I usually don't really get how it's going to be important for our real life struggles. But today's a lucky day because by the end of the video, you will finally have your biggest dream come true. Understand why an AI program that can predict the folding of proteins is actually a huge advancement for scientific and medical research. Don't lie. I know that's all that you wished for. You're welcome. Okay, so first things first, what are we talking about? Proteins. So not exactly the kind you're used to hearing by your local gym bros who only swears by dried chicken and unspiced broccoli, but the much, much broader and more beautiful word of protein that do a lot more action than just giving alpha men big muscles. Wow. Proteins are everywhere. They do so many things. There are some of your hormones, like insulin. They are used to send messages between the cells. There are also the receptors that catch the signal and understand the message. They are used for structure and support of your tissues and organs, for example, collagen or keratin. They are part of your immune system because they are your antibodies. They are used to facilitate or sometimes just allow chemical reaction to happen in your body. For example, you would not have DNA replication without proteins. They are also used for transport, for example, hemoglobin that transport the oxygen throughout your blood. They are so many applications, and proteins are really at the center of what makes a body function. So, yes, sports boy is right, proteins are important, but for so many more things that he will never imagine. Now, we have to dig a little bit deeper into what proteins are made of to understand why this Nobel Prize is really cool. So you can imagine a protein kind of like a pearl chain, with every single pearl being one amino acid. In our body, we have 20 different amino acids, and there are 11 that we can make ourselves, and there are 9 that we cannot make that we have to get from our food. So, eat your proteins, kids. The order of the pearls, or the order of amino acids, is called a sequence, the sequence of the protein. But the thing is, in your body, this kind of long string doesn't stay like this, like a lifeless worm wobbling around. It's twisted, it curls on itself until it reaches its final form. And its final form is as important as its sequence to give it its function. Without a proper form, the protein cannot work. This is actually the explanation behind the prions disease. The people who know about prions disease are currently hyperventilating in the back, but for those who are not aware of what it is a very, very serious disease where one kind of protein in your brain gets misfolded, so it has the wrong shape, the wrong arrangement on itself. And because it is misfolded, it cannot do its job properly, but also it will misfold all of the same proteins that are around it, so you can understand kind of how the contagious effect works. And this causes huge neurological damage. It causes something that we call spongy brain because your brain gets holes or plaques, and it is incurable. If you get it, eventually you will die. So this is how important the structure of a protein and the way it folds is. The thing is, figuring out the shape or the folding of the protein is for now, a very, very complicated process. Because knowing the sequence is quite easy. But to understand the folding of the protein, you cannot just look at the sequence of amino acid and predict how it's going to fold. You have to look at them. But have you ever seen a protein? No. They are teeny tiny, and so to study them, you need very, very powerful techniques called X-ray crystallography or cryo-electron microscopy, and they are super long, super burdensome. You need to have the protein purified, which means you cannot have any other proteins around it. You need to bring them to very, very low temperature. It is a whole process, and so far, out of the many, 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 many proteins we know, a few millions, a few hundred millions actually, we only have 170,000 that we know the structure of. So now you might tell me, okay, but like, who cares? We don't know how proteins are folding. None of my problem, they fold by themselves. Just leave them alone. Why do scientists have to know everything? And on one hand, you could be right, but on the other hand, first, knowing for knowing is very cool, and you never know when you will need something. And second, no, you didn't see that argument coming, right? I'm gonna explain it, don't worry. So, why no, young Padawan? It is because most of the drugs, and I'm talking about legal drugs, but also illegal ones, are molecules or proteins that did not exist before, that we created. Imagine a cell, like a cancer cell. It takes signals from its surrounding. And in that specific case, there is a molecule that is floating around that, if it is received by the cell, will tell the cell to proliferate. And so in that case, what happens is that the cancer cell has a receptor, which is kind of a tiny arm with a keyhole, that can receive one specific type of molecule, so the key. And when that key gets into the keyhole, everything is activated and the cancer cell can proliferate. Now, imagine you want to stop that proliferation. What you could do is create a protein that would fit also into that keyhole better than the original signal molecule, and that would not trigger the proliferation. But how do you create it now? Well, what is happening currently is that there are a few programs that can kind of guess the folding of a protein, but what we are currently doing is that we are taking the original signal molecule, we are studying its sequence, and then we are replacing one by one some amino acids to make a new sequence. We let the protein fold by itself, and then in a lab, we test every single one on the receptor. It is so long, it is so burdensome. As you can imagine, we cannot test every single combination. There would be way too many. So now imagine that there is a program that you can give the receptor structure to, so the keyhole structure, and it would create a sequence of amino acid and fold it by itself and figure out if it's going to fit the keyhole or not. That would be amazing, right? Because now that program could test thousands, millions of combinations, even one we hadn't thought about, even one that are very different from the signal molecule. You never know. And it could do that in a very short time, and then you would take the best candidate and you would test them experimentally. So something that used to take months could now be done in a few hours. So as you can understand, the goal is absolutely not to replace structural biology, so the field that looks at the structure of protein, but to make it better, faster, stronger? For those who are still a bit confused on how such an algorithm can be built, the theory is actually quite easy. The coding is a pain in the ass, however, obviously. So the process of teaching that kind of algorithm, how to do it, is called deep learning. So how you do it is that you give your program a set of sequences of proteins and their respective folding, their respective 3D structure that you are sure of it. And you make the program learn by itself, okay, this kind of amino acid will fold like this, and so it will give this structure. And you do that for thousands of them. And then once this training process is over, you make sure that the program is accurate by giving it only the sequence of proteins that you, as a scientist, know the structure of, and then you see if the structure it predicted fits the structure that you know exists. If the program guesses right, amazing, you have a program that works. If it does not, you adjust the parameters of the code, and then you go again. Unfortunately, this kind of simplistic approach is too simplistic for the kind of problem that is folding of proteins. Who would have guessed? And so this specific algorithm, the original one that led the scientists to building the one that they got the Nobel for, created an extra step that is called distance matrix. And basically it's kind of a complicated thing, so I will not go into the details here, but it's a matrix that estimates how distant each atom should be from each other, each atom of the sequence should be from each other, based on the very basic rules of physics of which atoms are going to attract themselves and which atoms are going to repulse. Bam! Now you have a program that, by just looking at a sequence of amino acid, can predict the folding of a protein. Amazing, right? And so because of so many scientists actually found it so amazing, they decided to create a contest in 1994 that happened every 2 years, where scientists that were creating programs to estimate how proteins would fold were competing against each other to try to predict the sequence of proteins that were not known yet. But in parallel, scientists were doing their crystallography methods to figure out what the sequence was, so it was, you could not cheat, don't worry. Up until 2016, the results of the prediction were honestly not great, but in 2018 and 2020, there was a huge breakthrough by the team of DeepMind, which is the team that the Nobel scientists are from, and they completely crushed the competition with their program AlphaFold and AlphaFold 2 that I described a little bit earlier, the one with the distance matrix. So now they consider that the big issue of trying to predict the folding of a protein from the sequence could be resolved, which is actually a huge breakthrough for science. What is really cool, what I think very cool, is that for once, AI is not for once, because it has happened again, but you know what I mean. This time, with this Nobel, it really shows that AI can be put at the service of science, and it will for sure lead to, I hope, many scientific breakthroughs that I hope I will be able to discuss on this channel. And I am honestly very, very excited for what's to come. Thank you so much for watching. This was Science Breakdown. I hope you stick around and I hope you stay curious.